Hidden viral load. Why viruses persist in healthy people. Most people unknowingly carry viruses inside their bodies, even if they appear perfectly healthy. These viruses don't always cause immediate illness. Instead, they can remain dormant for years, sometimes decades, quietly replicating or integrating into our cells. A large-scale study recently published by researchers at Harvard Medical School has provided a detailed look into how common these latent infections are, which viruses persist the longest, and how our own genetics influence viral load. The scope of viral persistence. The study analyzed data from over 917,000 individuals across 3 medical databases, examining blood and saliva samples to measure circulating viral DNA. The goal wasn't to identify new pathogens, but to quantify the viral burden already present in seemingly healthy people. This matters because understanding baseline viral loads can help us predict future disease outbreaks and develop targeted treatments. Researchers found that viral load, the amount of viral DNA in a person's body, varies dramatically based on genetics, age, sex, and even lifestyle factors like smoking. The team identified 82 specific genetic locations linked to viral DNA levels, particularly within the major histocompatibility complex, MHC. The MHC is critical for immune response, so these findings suggest that our ability to suppress viruses is partially determined by our genes. Virus-specific trends. Certain viruses exhibit predictable patterns of persistence. For example, Epstein-Barr virus, EBV, becomes more common with age, while herpes virus HHV7 declines after middle age. Seasonal variations were also observed. EBV viral load increased in winter and decreased in summer. This suggests that environmental factors influence viral replication rates. Crucially, the study established a direct link between high EBV viral load and an increased risk of Hodgkin's lymphoma later in life. This is a significant finding because it implies that antiviral therapies could potentially reduce this risk, although further research is needed. Interestingly, the same link was not found for multiple sclerosis, MS, despite EBV being a known trigger for the disease. This suggests that the way the immune system responds to EBV is more important than the viral load itself. Implications for future research. The study highlights the importance of large-scale genetic biobanks for viral research. By analyzing massive datasets, scientists can uncover subtle connections between viruses, genetics, and disease. 3 common viruses, annuloviruses, were found in 80–90% of the population, but their role in disease remains unclear. It's also important to note that this study focused on DNA viruses. Future work should expand to include RNA viruses like coronaviruses, which operate differently. Moreover, ancient viral DNA embedded in our genomes may still influence our health in unexpected ways, adding another layer of complexity to viral persistence. This finding is an example of why virus research in large genetic biobanks is important, says Kamataki, emphasizing the need for continued investigation into the interplay between viruses, genetics, and the human body. The study reinforces that viruses are more prevalent than previously thought, and that individual susceptibility to viral diseases is shaped by a complex combination of genetics, environment, and immune function.